Hello guys, welcome to my channel. Today we are going to talk about the role of coenzymes in metabolism. Okay, so let's talk about coenzymes first. So except ribosome, all enzymes are proteinaceous in nature. We know enzymes are proteinaceous in nature. Many enzymes require the presence of non-protein substance for the catalytic activity. If the non-protein substance is organic, it is known as coenzyme. If it is inorganic, it is known as cofactor. Okay, so for the enzyme, okay, enzyme is proteinaceous in nature. For the enzyme to complete its action, it requires a non-protein substance, and if that non-protein substance is a organic in nature, then it is said to be coenzyme. If it is inorganic in nature, it is said to be cofactor. Okay, so this coenzyme or cofactor may be an integral part of the enzyme molecule, or its presence may be required during the reaction. Okay, the protein portion of an enzyme that requires a coenzyme is called apoenzyme. The apoenzyme combines with the coenzyme to form the holoenzyme, which is catalytically active form of the enzyme. Okay, now the coenzyme, okay, it may be integrally bounded with the enzyme, or its presence may be required during the reaction, okay, as a second substrate. Okay, so for example, if this is inactive enzyme or apoenzyme, when it binds with the coenzyme, okay, it forms the active enzyme or holoenzyme. Okay, so what these coenzymes actually are, they are the derivatives of B complex vitamin. Okay, the coenzymes generally contain vitamins of B complex family. Example: thiamine, riboflavin, niacin, pantothenic acid, pyridoxine, folic acid, vitamin B12, lipoic acid, biotin, etc. Okay, so some of these example, lipoic acid and biotin, they act as coenzymes by themselves, right? And the others, they need to be converted into the coenzyme form. So coenzymes are generally required in group transfer reactions. For example, oxidation reduction, transamination, phosphorylation, etc. In oxidation reduction, there is transfer of hydrogen. Okay, in transamination reaction, there is transfer of amino group. In phosphorylation reaction, there is transfer of phosphate group. Okay, so they can be divided into two groups on the nature of the chemical groups they have to transfer. First, the coenzymes involved in the transfer of hydrogen, and second are the coenzymes involved in the transfer of groups other than hydrogen. Okay, so let's talk about the coenzymes that are involved in the transfer of hydrogen. So what are the coenzymes that are involved in transfer of hydrogen? So FMN, FAD, NAD+, NADP+, lipoic acid, coenzyme Q, these all coenzymes, they act to transfer the hydrogen. Okay, they act as a carrier of hydrogen. Okay, for example, if FMN, okay, it accepts hydrogen, it will be converted into FMNH2. Similarly, FAD will be converted into FADH2. Similarly, NAD+ will be converted into NADH. Okay, so these coenzymes, okay, they accept hydrogen, okay, or they can get reduced and they can again get oxidized. So they are involved in the transfer of hydrogen. Now coenzymes involved in the transfer of groups other than hydrogens, they are TPP, that is thymine pyrophosphate or thymine diphosphate, TDP. They are involved in the transfer of phosphate group. And there is coenzyme A. Okay, coenzyme A, it is involved in the transfer of acetyl group. Pyridoxal phosphate, PLP, it is involved in the transfer of amino group. Okay, and so on. Okay, so these all are the coenzymes which are involved in the transfer of groups other than hydrogen. They transfer other different groups, right? So okay, now the chemical change in coenzyme is opposite to that in the substrate. Okay, in most of the cases, enzymes do not change themselves. Okay, they only, sorry, not enzymes, coenzymes do not change themselves. They only transfers different groups, and in the end they get regenerated. Okay, so Some coenzymes accept a chemical group from one substrate and donate it to another. They accept a chemical group and donate it to another. Thus, they only act as carriers and regain their original form at the end of the reaction. Okay. Good example of that is pyridoxal phosphate, which acts as a carrier of amino group in transamination reaction. Okay. We need to be familiar about this pyridoxal phosphate because it is a very important enzyme in protein metabolism. Okay. So this coenzyme, this pyridoxal phosphate, this coenzyme, it is a very important coenzyme for protein metabolism. Okay. So they are helping in transamination reaction. Transamination reaction is transfer of amino group, okay, from one amino acid to another keto acid, okay, and thus forming another different amino acid. You can see an example of transamination reaction here. By the enzyme, this glutamate oxalo acetate transaminase, okay, the amino group of aspartate is transferred to alpha ketoglutarate and forming glutamate. Okay, here this PLP, pyridoxal phosphate, is acting as a coenzyme. What it does, it accepts an amino group. Okay, so it accepts an amino group. I will show you with a pointer. Okay, so this pyridoxal phosphate, it is a coenzyme. It accepts an amino group, okay, from aspartate. Then pyridoxal phosphate will be converted into pyridoxamine phosphate because amino group has been attached here. This aspartate is converted into a keto acid called oxaloacetate. Okay. Now this pyridoxamine phosphate again donates this amino group to another keto acid known as alpha ketoglutarate. And when alpha ketoglutarate accepts amino group, it is converted into glutamate. Okay. After donating this amino group, it is regenerated into its original form, that is in the form of pyridoxal phosphate. Okay. So this is how coenzyme like pyridoxal phosphate, they are involved in this transamination reaction, or they are involved in transfer of amino group. Okay. So sometimes the change in the coenzyme is more important than the change in the substrate. In the previous example, we just saw that change in coenzyme, that is not important. Coenzyme, after all, it gets its original form. Okay, but sometimes the change in coenzyme is more important than the change in the substrate. Okay, for example, in aerobic glycolysis, we all know in aerobic glycolysis, glucose is converted into pyruvate, and in that process, NADH is produced. That is again reduced in one of the reaction. Okay, the reduced NAD plus transfers its hydrogen atom to oxygen, forming water, and NAD plus is regenerated. Okay, so in glycolysis, when glucose is converted into pyruvate, NAD plus is reduced in one of the reaction. That means NAD plus is reduced into NADH. Okay, now reduced NAD plus or NADH, we have to understand reduced NAD plus means NADH, it transfers its hydrogen atoms to oxygen, forming water. And when NADH transfer its hydrogen, NAD plus is regenerated. Okay, this occurs in aerobic condition. Here NADH is releasing its hydrogen atom to oxygen in electron transport chain. You have to remember that. Okay, so this is an aerobic condition. In aerobic condition, NAD plus is regenerated through electron transport chain. Okay, in the mitochondria. Okay, so but in anaerobic condition, NAD plus cannot be regenerated due to lack of oxygen, right? So what happens, therefore an additional reaction occurs in which pyruvate is reduced to lactate and NADH is oxidized to NAD plus. Okay, now this is a very important reaction. Okay, here the change in this substrate is very important. NADH in aerobic condition, it is regenerated through TCA cycle, but in anaerobic condition, it cannot be regenerated through NAD plus. Okay, in aerobic condition, it is regenerated by electron transport chain, but in case of anaerobic condition, this NAD plus cannot be regenerated by electron transport chain because there is lack of oxygen. In that case, what happens, there additional reaction will occur where pyruvate will be converted into lactate, and in that conversion, okay, that is a reduction process. Okay, pyruvate is reduced to lactate. In that reduction process, NADH will be oxidized to NAD plus. Okay, so here regeneration of NAD plus is more important for continuation of glycolysis. Okay, So you can see this reaction. Okay, this is the glycolytic reaction. Okay, here is the production of NADH. Okay, this in aerobic condition, this NADH will be regenerated as NAD plus by electron transport chain. But in anaerobic condition, okay, there is the conversion of pyruvate into lactate, and in that process NADH is regenerated into NAD plus, which will be useful for the continuation of glycolysis in anaerobic condition as well. Okay, so thank you guys. Please like and subscribe my channel.